Megan Vega, and this is my video for the DNA Replication Lab assignment. In front of me I have the printout from the slideshow, as well as my notes that I'm going to use to describe the process of replication from the lagging and leading strand. So as I hold up this picture, I labeled everything where it belongs, and then I'm going to explain the process. So replication occurs in three major steps: the opening of the double helix and the separation of the DNA strands, the priming of the template strand, and the assembly of the new DNA segment. During separation, the two strands of the DNA double helix uncoil at a specific location called the origin. So first, let's talk about initiation. During initiation, the DNA strand must be open to allow access to the base pairs on the template strand. Initiation begins as helicases break hydrogen bonds between the base pairs and uncoil the strands. This turns two strands into separate single-stranded molecules. One is found at each origin of replication. Since the strand is no longer attached, supercoiling becomes an issue. Topoisomerase fixes this by removing the tension and then reattaching the DNA. As the helicase is separating the two strands away from one another, the one single strand and the other single strand are complementary to each other. To prevent them from reforming, the single strand binding proteins attach to the two single strand complementary pieces of DNA. This prevents them from coming into contact with one another. DNA polymerase III then adds in the new nucleotides to the growing DNA strand. It also matches complementary base pairs to create a daughter strand from the one parent strand. It only adds to the 3' end and never the 5' end. Primase is the enzyme that helps the polymerase get started. Since the strands are anti-parallel, a DNA polymerase III is needed on both ends of each strand running in opposite directions. This creates leading strands, lagging strands, Okazaki fragments. Okazaki fragments are when nucleotides are unable to connect together through bonds. DNA ligase recognizes these sections in between Okazaki fragments that are not connected. It attaches and creates the covalent bond needed to turn two adjacent Okazaki fragments into one larger piece of DNA. Since you cannot attach the new Okazaki fragment to the beginning of the previous one, DNA polymerase I will go and remove the old nucleotides and replace them with new DNA. Next, I'm going to go over this picture from the slides that were provided. And so number one, I'm going to point out where the 5' end is. So you can see it's right there. And then they're opposite of each other, so the other one is going to be right there. Where would the helicases be located? And I said that it was located here, and it is moving in the opposite direction of the new strand, so it is lagging. So number 3, is the new strand a leading or lagging strand? The new strand is lagging because it is traveling the opposite direction of the helicase. Therefore, it will be doing DNA replication in short lagging sections.